Assalamualaikum and hi. My name is Muhammad Aminuddin bin Zainal. In this video, I would like to represent to all of you my assignment about DNA and RNA molecules and structures and functions about plasma membrane. DNA and RNA is a nucleic acid that consists thousands of nucleotides called polynucleotide. Nucleotide is a building blocks for nucleic acid that compose of C, H, O, P, and N. There are two types of nucleic acid which are DNA and RNA. Nucleotide consists of pentose sugar, nitrogenous base, and phosphate group. There are two pentose sugar which are deoxyribose and ribose, while there are two nitrogenous bases which are purine and pyrimidine. There are two pentose sugar which are ribose and deoxyribose. Ribose sugar is a pentose sugar for RNA, while deoxyribose is a sugar for DNA. How do we recognize it? By looking at the second carbon atom, which RNA has only OH atom, while deoxyribose has only hydrogen atom. Nitrogenous base consists of two, which are pyrimidine that has single ring and purine that has double rings. Pyrimidine consists of cytosine and thymine for DNA and uracil for RNA. Purine consists of guanine and adenine. As you can see on the slide, these are the DNA based on Watson and Crick model. DNA consists of two polynucleotide chains. There are two strands that twisted and antiparallel to form double helix. Both strands are joined together by phosphodiester bond. Each strands are made up of deoxyribose and phosphate group, repeating to form sugar phosphate. Both DNA and RNA has their own uniqueness, which we can differentiate them. On the slide, as you can see, there are 8 differences, which are pentose sugar for DNA is deoxyribose, while pentose sugar for RNA is ribose. Other than that, DNA form a double helix, while there is no double helix formed in RNA. Lastly, DNA consists of adenine, guanine, cytosine, and thymine, while RNA consists of adenine, guanine, cytosine, and uracil. I choose these 3 points because it is general and it is easy to memorize it. On the last slide for this chapter is about RNA. There are 3 types of RNA, which are mRNA, or we call it as messenger RNA, tRNA, or we call it as transfer RNA, and the last but not least is rRNA, or we call it as ribosomal RNA. Each types of RNA has their own uniqueness and functions. Okay, for the next chapter, I will talk about structures and functions of plasma membrane. Plasma membrane is including in endomembrane system. Endomembrane system consists of nucleus membrane, endoplasmic reticulum, Golgi body, lysosome, vacuole, and plasma membrane. Plasma membrane separates the living cell from non-living surrounding. Size for the plasma membrane is 7.5 until 8 nanometer thick. This plasma membrane has proposed by Singer and Nicholson and rebranded as fluid mosaic model. Fluid word in the terms is referred as phospholipid can generally move laterally or flip-flop in rare cases. Mosaic word in the terms referred as there are proteins that embedded within the phospholipid bilayer. Phospholipid consists of head and tail. Characteristic for head are polar and hydrophilic, while characteristic for tail are non-polar and hydrophobic. Hydrophilic means phospholipid head is attracted to water, while hydrophobic means tail is avoiding the water. Between the phospholipid bilayer, there are hydrophobic regions formed by phospholipid tail. There are two types of protein at the phospholipid bilayer, which are integral protein and peripheral protein. Integral protein is a protein that partially or fully embedded within the phospholipid bilayer, while peripheral protein is a protein that attaches loosely to the surface of phospholipid. Lastly, membrane plus protein has their own functions, which are it acts as transport protein. Other than that, it acts as receptor and acts as enzymes. Besides, it acts as cell recognition and as an attachment site of cytoskeleton.